Hello everybody, this is the review video for our first genetics test. It is currently 3 a.m. on Monday as I'm recording this, and so I'm not going to fix any mistakes I make, and I'm going to try not to take too long. So if you feel like I'm rushing, please go back and look at my other sets of notes, look at the other videos that I've posted, both that I've made and that are professionally made, and I'm sure you'll get your questions answered through that. So let's start with the history of DNA. You don't need to know the exact names and exactly what happened in each of the experiments, but you should expect to see a couple of them on the test, in which you'll be presented with a description and asked to make a prediction or analyze, what happens if I did X or Y differently. So being familiar with them will be helpful. So starting with Griffith was the one who realized that there was an actual chemical that was containing the genetic information or the information about how to make an organism. Before that, they didn't really know that it was an actual physical molecule. And he figured that out because when he took different strains of bacteria and combined them, these interesting things happened. So starting out with a rough strain of bacteria injected into a mouse caused it to live. When he injected a different strain of the bacteria, the smooth strain, which happened to have this little yellow capsule on the outside, the mouse died. And that was because the bacteria's capsule protected it from the mouse's immune system. And so the bacteria didn't die from the immune cells and managed to kill the mouse. When he destroyed the smooth strain, the mouse lived, and that makes sense. It's a dead bacteria. However, when he combined the smooth that was killed with the rough, the mouse died, and that's because DNA from the smooth strain was actually picked up by the rough strain, and it caused the rough to actually make a capsule and therefore protect itself from the mouse's immune system. And so what happened there was actually transformation. And that word should sound really familiar now, because when we were in Madison, you did a bacterial transformation. You took DNA from one organism, I forget if it was a fruit fly or a firefly or what it was. When you put it into a bacteria, you transformed it. So the whole thing proving that there is a chemical that contains the information for. So Avery, McCarty, and McLeod went off to show that the chemical molecule was actually DNA, not proteins. Remember, they thought that it was proteins because proteins are made of 23 different kinds of amino acids, thereby allowing for lots of different combinations to make the stuff of life, whereas DNA only has 4 different monomers, which are nucleotides, the different A's, T's, C's, and G's. So how did they prove that it was DNA? So they took some heat-killed S cells and split it up into 3 groups. One of those groups had DNA and RNA, but no protein. One had proteins and DNA, but no RNA. And then one had RNA and proteins, but no DNA. Then they added rough cells to that mixture of heat-killed S. That's what this R cell thing is. And when they did that to this group, this first group, they found that S cells appeared, or in other words, those R cells had been transformed and appeared to have this capsule on them, so they looked like S's. And so this suggests to us that something here, either DNA or RNA, was passed. We know that it was DNA, but they didn't necessarily know that. Then they added rough to proteins and the DNA. Once again, S cells appeared. Of course, it's because the DNA was there. And then in the last experiment, no S cells appeared because there was no DNA. There was some confusion about the protease and RNase and all of that, so just go over that real quick. They should do the experiment twice because people were convinced still that it was proteins that was doing this, and said, well, maybe it's because DNA is wrapped around proteins. And so in this case, for example, the S cells appear because there's actually a little bit of protein stuck in that DNA. So to prove that wasn't the case, they added proteinase or protease to destroy any leftover protein just in case. And same idea here, but the results were the same, thereby letting them make this conclusion that DNA is their hereditary molecule. So Hershey and Chase actually had the exact same question as Avery, McCarty, and McLeod. They just answered it in a different way. This is the blender experiment. What they did is they grew viruses in different chemicals. So in one situation, they grew it in sulfur. Remember that sulfur is one of those compounds that's not found in many of the different macromolecules, but it is found in protein, in a few proteins. It's in some of the amino acids of proteins. And so when they let these viruses duplicate in material containing sulfur, those viruses took up the sulfur into their little protein coats. And this like little pink, where's my pointer? This little pink highlighting here is showing the protein coats with that sulfur in it. And why did they bother doing this? It's because they couldn't see the viruses. They are too small for us to see. But by using this radioactive sulfur, they could trace where where the proteins were moving. So they let them divide. Because viruses have this protein coat on the outside of them, they could follow it. So then they let the viruses infect bacteria. The viruses injected their little DNA inside of the cells, and later on, when they separated the bacteria from the viruses, they could see on the top where the viruses were this pink stuff. And they know that viruses are less dense, so they're floating on top, whereas the bacteria are floating in the bottom, and there is no radioactivity there. So this has shown them that because they knew that the sulfur and the proteins were on the outside and nothing radioactive ended up on the inside, that proteins must not be the stuff of life, because if it was, then the viruses would have need to put it inside of the bacteria. Over here, we've got they radioactively tagged the DNA with phosphorus, because phosphorus is part of DNA. It's sugar phosphate backbone, right? And when the viruses infected the bacteria, they inserted their DNA. Once again, they separated them out and saw that the bacteria outglowed, had this radioactivity, because the tagged DNA was inserted into them. Hopefully that made some sense. So Chargaff is the one who discovered that there are equal ratios of adenine and thymine and cytosine and guanine in DNA, which now we know that's because adenine pairs with thymine, like chemically they fit together, and guanine pairs with cytosine. This is what his data looks like. I wouldn't be surprised if you saw this on the AP test. I think it's on my test. So let's take a quick peek at it. So each of these different lines represents like a different organism. And you can see here that, for example, when I have 28% of the DNA being adenine, 27% is thymine. That's pretty close. It's because they pair together. Whereas guanine and cytosine are pretty equal. So if you look at all those different individuals, you'll see that the percentages of A's and T's is about the same, and the percentages of C's and G's is about the same. Finally, Watson and Crick used Rosalind Franklin's data of exocrystallography to discover the double helix shape of DNA, two sugar phosphate backbones with the the nitrogen bases facing to the inside. There's more to know about this. It's fascinating. I'm going to see if I can try to fit it in at some point, if not before the AP test, whenever that happens to be. Maybe we could do it later on. All right, let's get into nucleic acid structure, the structure of DNA. So you should remember that prokaryotes have circular chromosomes, and they also have tiny plasmids, which are extrachromosomal. They don't contain essential information for life, whereas eukaryotes have linear chromosomes. So here's a peek at that circular DNA. pointer. Circular DNA, small plasmids, where a eukaryote has these linear, straight pieces of DNA. The structure of a nucleotide consists of the following: 1 phosphate, 1 sugar, and 1 base. And this goes for DNA and RNA. They all have nucleotides as their monomers. There are some differences between the bases and the sugars. We'll get to that in a second. So the types of bases for DNA and RNA can be classified into 2 groups. There's purines and pyrimidines. Purines are adenine and guanine, and they have 2 rings in their bases. Right here, 1, 2, adenine, guanine. Pyrimidines, on the other hand, have only 1 ring, and cytosine and thymine and uracil are all pyrimidines with 1 ring. If you get those mixed up, purines versus pyrimidines, please look back at your notes for some tips on how to remember that. So what's the difference between DNA and RNA? DNA has a different kind of sugar. It has a deoxyribose sugar versus RNA's ribose sugar. If you look at the chemical structure here, you will see that they are just a little bit different. RNA has adenine, uracil, cytosine, and guanine as bases. So it's got U versus DNA, which has a T, thymine, instead of uracil. Don't ask me why. I've tried to look into it, and it's something that would have occurred so long ago in the evolution of life that I don't know that we really know. Also, RNA is single-stranded, DNA is double-stranded. So those are the 3 differences between DNA and RNA. Little romantic joke for you. All right, so one of the last things we need to know about DNA is the bonding. Remember that on the sugar-phosphate backbone, there are covalent bonds between the different components, whereas between the nitrogen bases, I have hydrogen bonds. These are relatively weak. They can be broken easily for transcription or for DNA replication. Also, the cytosine and guanine form 3 bonds between them, and adenine and thymine form 2 hydrogen bonds. I don't know that that's important, but just in case, I'll tell you anyway. All right, directionality. So hopefully we get this by now. DNA and RNA are DNA is anti-parallel. The top of one is going to be a 5' versus the other top is going to be a 3', and they're anti-parallel because the kitty corner side is going to be the same. What makes them a 3 or 5'? You can study that a little bit here, kind of looking at the picture, but it's not important for the AP test. All right, then let's jump into protein synthesis. So steps of protein synthesis are transcription and translation. Both bacteria or prokaryotes and eukaryotes have to do protein synthesis. The difference between the 2 types of cells is that eukaryotes will have RNA processing, and this can occur because these cells have a nucleus, and the nucleus is separating the DNA from the ribosomes where translation is going to happen. Over here, the DNA, because it's a bacteria, doesn't have a nucleus, it's just floating free in the cytoplasm, and the ribosomes can start to translate that RNA as soon as it's produced, thereby leaving no time for this processing for things to happen with that RNA before it leaves the nucleus and goes to be translated. So the overall process is looking like this. Here's my DNA. I'm going to be splitting it into 2. I'm going to transcribe it into RNA. Those RNA bases are going to be read 3 at a time. 3 bases are called a codon, and each of the 3 letter base combinations is a code for 1 single amino acid. And so I end up with this chain of amino acids that becomes my protein. So DNA to RNA is transcription, RNA to protein is translation. Just some reminder about some of the features of the genetic code system. It is redundant. What that means is that I can have several different codons that will all code for the same amino acid. What that means for mutations is that if I change one of these letters here, I will still end up with the same amino acid. Sorry, if I change the last letter here, I'll still get the same amino acid. The genetic code is also unambiguous. What that means is this codon, for example, ACU, is unambiguous because it only means threeing, I think that's what that's called. It never means anything else. So it's perfectly clear as to what it means. Just like in English, cat always means this thing. That's a cat. I watched a really funny video today of cats pushing things off of shelves. If you need to laugh, you should look for that. So cat always means this, but kitten also means that, kind of. So redundant, two words can give me the same organism, but unambiguous, both of those words only mean this thing. Remember, the genetic code is also universal, meaning that I can have a gene in one organism, take it out, put it into a different organism, and it should still be able to express itself, or could still be able to be expressed. And we can see that in the situation where we've got firefly genes put into either a plant or a pig, and look, they're being expressed. I think that's just amazing and powerful evidence of evolution. So let's get into transcription really quick. Transcription starts with the RNA polymerase right here. This is our one enzyme in transcription, is binding to my promoter. So the promoter is this light green area right here. The promoter is the only region where the RNA polymerase can bind. So it binds, it in this first step is initiation, it's binding, then it is going to work on elongation. So it starts to, the RNA polymerase starts to separate those strands, and as it separates, it's going to build on my new RNA. Notice it does not stay attached here, and this, you probably knew that at the time, but now that you know what DNA replication is, sometimes kids get confused and think the RNA stays attached to the DNA. It doesn't. That's not the point, right? The RNA is a messenger. It is taking the information from the DNA and taking it to a different place. So first step, initiation, then elongation, and then termination. Once the RNA polymerase reaches a signal down here, be it a termination sequence in bacteria or in eukaryotes, there's a different sequence that you don't need to worry about. Point is, it gets to a place where it knows to stop. The RNA polymerase falls off, and my RNA is finished to either be, it can be processed, or if it's in the bacteria, it can go right to being translated. Just a little closer look at initiation. These little purple grape-like things are transcription factors. Remember, those are proteins that help the RNA polymerase to bind to the promoter. Oh yeah, also remember that transcription happens 5-3. So I've started to build this 5, and I'm working towards the 3. So in this case, my RNA polymerase is moving in this direction. Remember that DNA replication also builds the 5 first and then works towards the 3. But what is this end right here? This side is a 3', right? I think you can see it over here. There's a 3'. So it just, I always think you build 5-3, but what that means is that I started on the 3' end of the DNA. Also recall that only one side of the DNA is transcribed, so that's the template strand. The non-template strand is not transcribed ever, so it's only the template one. And then just a reminder about the nitrogen bases and how that works here. So on the DNA, if I have a thymine, the other side of the DNA would have been an adenine, right? But on the RNA, it's the same, yeah? Now, when I have an adenine on the DNA, the other side would have been a thymine on the DNA, but because RNA doesn't have thymine, I get a uracil instead. So if you look, kind of compare these for a second, the RNA to the DNA, you'll see they're identical, except that whenever I have a thymine on the DNA, I will have a uracil instead. So then after I'm done transcribing, I can go into splicing or any other sort of modification that I want to do with the DNA. 5' caps and poly A tails are modifications. You don't need to know those for my test, but they could potentially be in the AP test. They should explain them and give you context, though. Splicing is the only one that we've talked about. Let me write that out. Splicing is when the introns, which are non-coding sections of DNA, are cut out. Why they don't code is a long story, and we don't have time to get into that. But basically, what matters is the exons, which are the coding sections, are stitched together into a new piece of RNA that is shorter. Recall that there are different ways of splicing. That's called alternative splicing, that can give me different kinds of proteins, which is why, even though we may only have, let's say, X number of genes, we can have Y number of proteins, which is much greater than the number of genes, and that's partially because of splicing. It gives me all kinds of new combinations, new variation. I think we're at translation. Yep. Here we go. So in translation, my RNA has floated out to the cytoplasm, and it's becomes attached to a ribosome. So first, the lower subunit of the ribosome binds to the RNA, then the upper subunit binds. And what happens here is the RNA is getting fed through the ribosome from a 5 to 3 direction. So it's getting pulled through here. And as it does that, the ribosome is reading the RNA 3 bases at a time, so 1 codon at a time. And whatever the codon is, let's zoom in a little bit here. Whatever the codon is going to determine which amino acid is going to be brought by our friend's tRNA. So tRNA stands for transfer RNA, and the transfer RNAs are going to bring 1 amino acid at a time. So here, for example, the codon is GGC. So the anticodon is going to be CCG. Anticodon on the tRNA, codon on the messenger RNA. So we're bringing in these tRNAs one at a time. There's 3 sites in the ribosome. One site here is where a tRNA is already leaving. See, it's already lost its amino acid and it's on its way out to go pick up a new one. In this middle site, the amino acid is currently being dropped off. And then in this first site, the amino acid is still attached, hasn't landed yet. So if I, for some reason, always think about like kids being picked up and dropped off from school. So over here, we've got a car going to pick up small child, and then car taking the kid to school. Not anymore though, boo. And then mom is driving up to school, hasn't parked yet, right? Over here, mom's parked, kid is still in the car but about to get out. And then over here, mom has dropped off child and is on her way home or wherever she's going. Right, so that is translation. So as translation is occurring, recall that the proteins are automatically folding all by themselves spontaneously. And that is in relation to which amino acids have been placed into the gene. So just recall the different levels of protein structure. So the first level is the order of the amino acid. That's determined by the letters of the DNA, of course. Then the proteins will right away start to either form this alpha helix, like a little spring, or a beta pleated sheet. And that will be also according to the amino acids. Then we've got the third level of protein structure, which is, to me, the most interesting one. And this is determined by the R group of the amino acids. So you might remember amino acids have essential carbon, and they've got an NH2 group. I forget what it is. They've got a carboxyl group. I think there's an oxygen over here too. And then I think there's a carbon up there. And then they've got this R group. This is the important one. This is the one that changes in all the different amino acids. And whatever is happening in that R group is going to determine how the tertiary folding occurs. So for example, let's say that this end is let's say this end is positive and this end is negative. Well, then it makes sense that they're like next to each other because they're folding in. Let's say this amino acid right here is hydrophobic. This amino acid here too is probably hydrophobic, and they're probably hiding there in the middle, kind of away from water. So whatever amino acid I've placed in that chain during the translation process is going to determine how this folding is happening. And again, it's spontaneous. Nothing is doing the folding for it. It happens all by itself as translation is occurring. This last level of protein structure would occur somewhere else, actually. And specifically, it can occur in either the ER or the Golgi apparatus. So post-translational modifications involve lots of different things. This is a cool graphic I just found on Khan Academy. I don't recall seeing this before, but I think it's really helpful. So some protein synthesis happens in ribosomes that are floating free in the cytoplasm or the cytosol. And those proteins can be sent right out to wherever they need to go. So they're going to have little tags on them that will tell them should they go up to the mitochondria and be used. Maybe there's an enzyme that's been produced that I need to have in cell respiration, so I'm going to send it to the mitochondria. Maybe it's an enzyme that's used in photosynthesis and it needs to go to the chloroplast. Maybe it is DNA polymerase or RNA polymerase and I need to send it to the nucleus. So some of those proteins are produced out here because they're going to be used in these different areas. Some of them will actually, they're being produced in the ribosome will kind of flow over to the ER and form rough ER. And those types of proteins tend to be ones that are going to be used either on the outside of the cell, so they need to be sent out. Let's say like insulin that's produced in the pancreas, it needs to be sent out to the rest of the body. Or it could be, let's say, a protein that's going to be needed in the plasma membrane or elsewhere. So again, the ribosome lands on the ER. As the protein's being produced, it's hydrophobic. Yeah, hydrophobic, and actually kind of ends up kind of stuck in the ER. From there, the ER will do some different modifications to it, send it off to the Golgi apparatus, which can do some more modifications, and will kind of package it, ship it, send it out to one of these different places. Couldn't get a big graphic on both spots. I just wanted to reiterate this. There's my nucleus, that's where transcription happened. RNA came out, was translated at it was translated at a ribosome. Ribosome landed on the rough ER, produced the protein. I think that's it right there. And notice how it looks different here than it does over here. So what that's showing is how during this process of moving through the Golgi apparatus, it's getting modified and kind of like modified all the way through, right? Looks different in each spot. And here in this case, this is a protein that's going to be used outside of the cell, and so I've sent it out of the cell membrane. All right, so that's protein synthesis. First step, transcription, DNA to RNA, build 5-3 using my RNA polymerase, the one enzyme and the only enzyme used here. Then I've got processing or splicing. I send my RNA out of the nucleus through the nuclear pore, if it is indeed a eukaryote. Then in translation, my ribosome is going to read the RNA, again in a 5-3 direction. Reading the RNA 3 bases at a time, or 1 codon at a time. Based on whatever that codon is, I will bring the appropriate amino acid. Notice these different shapes here representing the different kinds of amino acid. And this will start to fold up to become my protein, could go off to ER and Golgi body to be further modified or not. Okay, next we've got gene regulation. So we'll start out with prokaryotic regulation. prokaryotes have a different reason for regulating their genes than eukaryotes do. So prokaryotes need to regulate their genes because their environments change really quickly. Let's say they came across a new food source, then they're going to need a way to digest that new food source, and they need to make some enzymes to digest it. But if that food source isn't present, they're not going to make the enzymes to digest it. That would be a waste of energy. One way to not make those enzymes would be through feedback inhibition, which we've talked about before. So that's where the final product that I make actually serves as a competitive or a non-competitive inhibitor and prevents this whole process from occurring. So I shut off this enzyme and keep it from working, and therefore shut down the assembly line. This is an okay way to do it. The problem is I still have already kind of produced some of these enzymes to start with, and that's a waste. Whereas over here, if I can just shut down the DNA that produces these enzymes, then I can save resources and not have to ever start the process to begin with. So how do bacteria regulate their DNA? They do it using operons. An operon is a group of genes right here that all kind of work together. Remember, the promoter is where the RNA polymerase is going to bind for transcription. The operator is where the repressor protein is going to bind. We'll get to that in a second. The regulatory genes are, here it is. There's my regulatory gene, the instructions for making a repressor protein. And then structural genes are the genes of interest, or the specific genes that I'm trying to manage here, the ones that I'm trying to turn on and off. So what does it look like when a bacteria has a gene turned on? So in that case, they're going to have their RNA polymerase bound to the promoter, and it's going to move along and transcribe, just like we just talked about, transcribing to make my RNA, and then it'll translate at the ribosome and make all the different proteins I need. So point here is, what does this look like? Well, it looks like RNA polymerase bound. No repressor is doing anything to interfere with that. When a gene is turned off, it will look like this, with a repressor bound to the DNA, right? And I can't make my RNA. Over here, no RNA is being made. So forget this little figure attached here. The point is just this the point is this repressor is bound and preventing my RNA polymerase from working and doing its job. So there's two types of operons. One type is repressible. Repressible operons are usually on. They can be turned off. That's why they are repressible. I can repress them. So when they're on, they look something like this. With the RNA polymerase bound, the repressor is not bound. The RNA polymerase is moving along and transcribing. This specific picture is showing the trip operon, and when the bacteria does not have tryptophan present, I'm going to need to make it. And so that's what's happening here. Tryptophan isn't present, RNA polymerase is attached, I'm going to be making these things which actually go on to make tryptophan. However, when I already have tryptophan present, as I do right here, there's no need to make anymore. So tryptophan is the regulatory molecule. It is going to land on the repressor, which causes the repressor to undergo a conformational change, and it binds with the operator, preventing the RNA polymerase from properly binding here, and it cannot move on to transcribe. So this is repressible because it's usually on, and I can cause the repressor to land on it, as here, and I can't do transcription, just your hand. Inducible operons are usually off. We can turn them on. So in this case, we're looking at the lac operon. The repressor is usually bound, but I can remove it by having lactose present. So in the other one, the repressor is only off if I don't have the regulatory molecule, right? So go back a second and look at how there's nothing here in the in the repressible operons. Whereas in inducible, the regulatory molecule causes the repressor to unbind. So usually off looks like this, right? The repressor is usually bound. RNA polymerase can't transcribe. I can turn it on. I can induce it by removing the repressor. In the case of the lac operon, what's happening is there is no need to make these enzymes to digest lactose if I don't have lactose. So when lactose is present, it removes the repressor in order for me to make specific enzymes to digest this. And you might recall that model that we used. The physical model had, I think, like a sword and a cube and something else that represented the different proteins that would use to digest lactose, which was, I think, your hand. So like the sword was the protein that was going to digest the the sword was the enzyme which was going to digest the protein. So let's take a look at eukaryotic gene regulation. So the purpose of eukaryotic gene regulation is pretty different than for prokaryotes. For eukaryotes, our big concern is differentiation and cell specialization. So we want to think about how can we go from one cell, which was made with sperm and egg, and cause it to become all these different kinds of cells. And how does an organism know where to make an ear versus a heart versus skin? And the answer is through gene regulation. So just a little quick visual here of what we mean by differentiation. So here we've got a liver cell, and livers need to be able to digest alcohol, and it does that using the enzyme alcohol dehydrogenase. So why does the liver make this? It's because it is allowed to transcribe this DNA, which makes RNA, which makes the protein alcohol dehydrogenase. It does not have, it does actually have instructions for making neurotransmitters, right? All of your cells have a complete set of DNA, except sperm and egg. So your liver cells have neurotransmitter genes, yet it doesn't make them, right? This is turned off there. Neurons then would have that neurotransmitter gene turned on. It'd make that RNA, it will make those different kinds of neurotransmitters, but it will have the alcohol dehydrogenase gene turned off. And this is how cells can be specialized and have different functions. Just a quick connection to cell cycle and mitosis. Gene regulation can be influenced by outside factors. So let's say I have a growth factor or hormone that lands on a cell and causes this signal transduction pathway to occur. And that inside of the nucleus here's my response to that signal transduction pathway. I've got transcription occurring and then translation of turning on this division promoting gene, which is going to cause proteins that will cause a cell to divide and move into mitosis. So lots of good connections there between our different units. So how exactly do eukaryotes regulate their gene expression? It can actually occur at any point from DNA all the way down to having a functional protein. So let's go through a few of those. First, it can occur in the packing and unpacking of DNA. So DNA can be really tightly packed as chromatin, preventing the RNA polymerase from binding, thereby preventing transcription from occurring, and I will not have protein synthesis. I can also control transcription by doing some things to make it difficult for transcription to occur. We'll come back to that one. I can also have RNA processing, like splicing, adding the caps and the tails. I can also control whether or not and how the RNA is actually transported out into the cytoplasm. I can degrade it using RNAi. Then for translation, I can do some different things to block ribosomes from binding to the RNA and prevent that from happening. Also, once my protein is made, I can damage it. I can combine different proteins. And that's all happening with different chemical signals. So lots of different spots here to kind of turn on and off these genes. And not all of them are going to occur on every single gene. What kind of control is happening depends on the kind of gene, what kind of cell it's in. Let's take a closer look at the control of transcription, because you might see this diagram on the AP test. How this happens is you've got another sequence here. This is another sequence called the enhancer sequence. So the enhancer sequence is going to allow a protein called an activator to bind to it. When the activator binds to the enhancer and the DNA bends with something called a bending protein, causes the DNA to bend like this, then that activator is going to actually get really close to this little green protein. This is called the transcription factor. Remember that from earlier? That helps the RNA polymerase to bind. So now I have this little complex. It's kind of confusing, but the point is I've got this complex of an activator, transcription factor, RNA polymerase. And all together, that allows the RNA polymerase to get really, really cozy and to start translation. If I don't have that activator present, it can make it difficult for the RNA polymerase to bind. And it might not bind at all, or it might bind just infrequently and cause transcription to not happen very often. So this is controlling the initiation of transcription. It is one common way of regulating gene expression. All right, let's really briefly review DNA replication, because I have a separate video on that. So replication of DNA is happening in a semi-conservative fashion. It does not happen conservatively or dispersively. Please recognize that. This is just kind of shown to say, well, how else could it occur if it doesn't happen semi-conservatively? Right, so remember that replication occurs differently depending on which strand we're talking about. So over here on this top strand, this is going to be my leading strand. We're right here. Try to follow that, because it gets confusing if you're looking at the other side. So over here, my top is going to be a 3, and my bottom is going to be a 5. And how do I know that this is a 3? Because if I'm going this direction, this had to be a 5, because I can only start with a 5. Recall that in order to start this process, I had to have a primer added. This picture doesn't show it because it's right there. It's off the picture, but I would have started with a primer first, and then my DNA polymerase, which here is shown in orange, is zipping along, attaching all of these bases. In addition to DNA polymerase working, I've also got helicase, which is kind of serving as a wedge, cutting the hydrogen bond between my nucleotides. And I also have topoisomerase working up here, which is relieving the twisting that happens as helicase is working. So leading strand, it's continuous. I form one long piece of DNA, and this is my whole new strand, ready and raring to go, so that I can go into mitosis. On the other strand, it's a little bit more complicated, because DNA can only be built from a 5-3 direction. And down here, my original strand, remember that's the dark blue, was a 5. And so that would mean that my new piece of DNA would need to start with a 3, as it is right there. Well, that's not going to work, right? So we can't go the same direction. This strand is not going to go into the replication fork. In fact, it's going to go the other direction. It's going to go out of the replication fork. So how does it do that? It starts with an RNA primer. Once again, my DNA polymerase will then build going this direction, as we have here. So here's my primer. It's a 5' primer, and then DNA polymerase building this direction until it gets to the end point, or it gets to another primer, and then the DNA polymerase will pop off. And we keep building these little fragments. They're called Okazaki fragments. And after those fragments are built, I'm going to cut out the primers. replace the primers with DNA and then stitch those different fragments of DNA together using ligase. Ligase, remember, works like a glue, gluing those different fragments together. And so that's replication. Now I have two identical strands. This one looks exactly like this one. It has the exact same bases, hopefully. This is one of my favorite cartoons, and I probably have to explain it to you. So topoisomerase, working like a little chiropractor, kind of doing the untwisting, getting out the knots. And notice his little oil or lubricant is helicase, because helicase, remember, separates the strands. So it's kind of like, loosening up the DNA. So many jokes in one, guys. Good stuff. All right, back to having possibly made a mistake. I didn't actually include a discussion of mutations in this review because I forgot until right now. But remember that we talked about point mutations. Point mutations are where I have one single DNA error. So instead of having, let's say, a T here, I substitute it for, I don't know, like a C. And that could cause it could cause a different amino acid to be produced during translation, thereby changing the protein. Please go back and review the different kinds of mutations. I think we have pretty good notes on that. But in order for us to not have that situation, it would be nice if we could fix the mistakes before they get translated. And so that's the point of this kind of proofreading. So there is, if I have a mutation, as I do here, a little enzyme called nuclease, which can actually cut out those different mutations. DNA polymerase will then repair it, and ligase will again stitch those different pieces together. So that's the process of repairing mutation. All right, let's move on to cell cycle and mitosis. So the cell cycle is just simply a cell's life. All cells go through the cell cycle. The main phase of the cell cycle is interphase, or I. Interphase consists of 3 subphases, G1, S, and G2. G1 is also called gap 1, and the point of gap 1 is to just grow and do normal cell activities. S phase stands for synthesis. This is when I will copy the DNA so that I can replicate the cell. And then in G2 or gap 2, I'm preparing to divide, copying all of the organelles for division. Lastly, I've got M phase or mitosis, which is the actual cell division. Remember that the cell cycle is regulated with checkpoints. And so each of these little red gates here represents the different checkpoints between the different phases. Those checkpoints could be internal chemical signals, they could be outside factors, whatever they happen to be. They're telling the cell when it should move on to one phase or the next. One other phase that the cell could potentially enter into is G0. G0 is kind of a pause. Some cells are permanently in G0, such as neurons or heart cells. Some cells will never enter that phase. So an example of an internal regulation of the cell is with cyclins and kinases. Remember that these are chemicals that the cell is producing that tell it, okay, it's time to move on, let's say, from G1 to S, and cyclin E appears to be one of those chemicals that helps that to happen. If you were confused by this, I think the cancer rating that I assigned you has some information about it. If you're still confused, I actually usually do the POGL on this, and so you get to spend a class period kind of exploring how this works. There's an excellent graph in the POGL. I do have the key up for the POGL, so if you have time, I know that you missed them, go ahead and take a peek at that. It's posted in the cell cycle folder. So anyway, internal regulation here. External regulation is another way that cell division or cell cycle can be controlled through different hormones. We can also control division externally through what's happening around the cell. So for example, let's say the cell is having trouble anchoring to a surface, as we have here. It may not divide. It shouldn't divide. It can also be controlled by how many other cells are around it. So if there isn't very many cells, like in this case, if I took some of these guys out, then these cells should be signaled to keep dividing and kind of fill in that gap. One reason why people get cancer is because the cells don't have a good sense of what's happening around them externally, or if they do, they're ignoring those signals and continuing to divide, let's say, knowing that they're not anchored to something that they need to be anchored to, knowing that there's already too many of them, but they just don't care. So cancer cells are not responding either to those external divisions, or they can have internal problems. There's lots and lots of different things that can go wrong with the internal chemicals, telling it when to go from which phase to the next. All right, really, really quickly, just some chromosome vocab. Remember that we have 2 sets of every chromosome as eukaryotes, and we see those 2 sets in all of our cells that are somatic cells. Remember, soma means body. So every single cell in your body and your cat's body and your favorite plant's body all contain 2 sets of chromosomes, except those cells which are germ cells or sex cells. So in the case of a plant, it's pollen. In the case of an animal, it's sperm or egg. Those only have 1 of each chromosome. And that makes sense because they're sex cells. We're going to combine 2 of them. Both cells have 1 set of chromosomes, combining to make 2 sets for a complete set of chromosomes for the organism to be healthy. Chromosomes, when they're duplicated after S phase, have some structures you need to know. The 2 sides, so here we've got 1 side of the chromosome and the other side of the chromosome, are called sister chromatids. They're held together by centromere. So normally in, let's say, G1 of the cell cycle, I have one copy of each chromosome, and then in S phase, I will duplicate them so that I've got 2 sister chromatids. This is considered 1 chromosome. This is also considered 1 chromosome. I often like to think of this as 2, though, because it's helpful when we get into mitosis or meiosis. All right, so G1, S, and then I would undergo mitosis. I would split up, and now I've got 2 separate cells with equal numbers of chromosomes as I had in the beginning. So we call those different sets of chromosomes homologous pairs. So 1 from dad, 1 from mom, 1 that's paternal, 1 that's maternal. They have the same kinds of genes in the same spot, although they could be a little bit different. So this, if it's a rabbit, could be the gene for straight ears, and this could be the gene for floppy ears, which in my opinion is the superior gene, because who doesn't love a bunny with floppy ears? So now when I copy the DNA in S phase, I will have the straight ear gene here and the straight ear gene here, because they have to be identical, right? Over here, I will have floppy ear gene and floppy ear gene, because they have to be identical. Still ear genes, right? But they can be different versions or different alleles. Main point is that homologous pairs can look like this, or they can look like this. They can be non-duplicated, or they can be duplicated. Here's a set of non-duplicated chromosomes. They must be in G1. And here is a set of duplicated chromosomes. They must have already undergone S phase, but not divided yet. So mitosis specifically is the division of the DNA, or the genetic material. Cytokinesis is actually what happens after the DNA has been divided, when I'm dividing the cytoplasm. So mitosis is going to give me 2 cells, and they're going to be identical. We call those daughter cells. So basically all that happens is the DNA is replicated in S phase. I line my chromosomes up in the middle of the cell, pull them apart. I've got two cells that look exactly the same. So the specific phases are prophase, prometaphase, metaphase, anaphase, and telophase. I was taught when I was a kid, I passed my algebra test. So I for interphase, right? That would have been G1, S, G2, whatever. And P for prophase, M for metaphase, A for anaphase, T for telophase. Ms. Corrado, you might be wondering, why did you not put in the extra P? And that's because prometaphase didn't actually exist when I was in high school. It was kind of described later on. All right, so what do the different phases look like? Here I'm finishing G2 of interphase. Notice how the DNA is all kind of messy. It's not condensed. The first phase of mitosis actually involves condensing that DNA. And why do we do that? Think if I asked you to divide a pot of cooked spaghetti into two, it's really tangly. It's going to be hard to equally divide that spaghetti into two different plates, let's say. So prophase, by condensing the DNA, allows us to evenly split it with one of each chromosome in each cell. Also during prophase, the centrioles are going to start separating, and I'm going to start forming this mitotic spindle with these little fibers that are going to be pulling on my chromosomes and pulling them apart. In prometaphase, those chromosomes are really starting to move towards the middle. They're not quite there yet, but they're moving towards the middle. And those fibers are starting to reach out and starting to get attached to the centromeres of the sister chromatids, getting ready to pull them apart. Also notice the nucleus is starting to dissolve. That's important because otherwise the fibers couldn't grab the DNA, right? Because they were stuck in the nucleus before. Metaphase, M for middle, so they're lined up in the middle. The fibers are now fully the mitotic spindle fibers are now fully and securely attached to the centromeres, and we're getting ready to do a little bit of a tug of war. Oh, also notice the centrosomes are kind of now securely at the poles of the cell. Anaphase, we're doing the tug of war and pulling apart the sister chromatids. They're separating. And it's really, really cool to watch this in a real cell. These little mitotic fibers kind of like, kind of wiggle back and forth and pull down into that centriole. I remember anaphase because to me this looks like an A. In the last phase, for telophase of mitosis, the nucleus is starting to reform around the chromosomes, and the DNA will start to decondense. So we'll go back to that kind of loose chromatin spaghetti-like structure. Also, my nucleus is going to reform. And then lastly, in cytokinesis, I start to get this little division between the cells. The cytoplasm starts to pinch off. Remember, cytokinesis happens differently in animal cells versus plant cells. In animal cells, it forms this little cleavage furrow, which I think looks just really cool. There's a little ring of fibers that's going to pinch off, versus in a plant cell where I get these vesicles kind of lining up and they form a new cell wall between the two cells. And that's how cytokinesis happens in a plant. Just a reminder that all cells reproduce using mitosis. We actually call it binary fission in prokaryotes, but it's the same concept, right? I copied the DNA, I'm going to pull them apart. True mitosis in eukaryotes is descended from binary fission in prokaryotes. Okay, we've reached the end. I hope that was helpful and not too painful to listen to. Please remember to post your questions in WebEx if there's anything that I can answer for you. If you are not comfortable posting in the group chat, you can chat me separately, but the group one would probably be best because if you have a question, other people probably do as well. All right, good luck, everybody.